Hi everybody, welcome to our next vodcast in our matter domain. In this one we're going to start getting into the cell. So here are things to keep in mind. What does internal cell structure look like? How do cells separate them from their external environment? How do cells communicate with other cells? And how do they communicate with their environment? So these are kind of the big picture ideas we're going to get from this vodcast and the next. So let's kind of backtrack a touch. So what do cells in general, obviously the basic unit of life, need to function and to stay alive? They need to be able to process matter. They have to be able to ingest, acquire, synthesize, whatever it may be. They need to be able to get matter because that's what we're made of. They need to be able to process energy. Okay, so in order to process the matter, you have to put energy in. But in order to get the energy out of the matter, that's kind of necessary for the cell to function. Everything needs energy to work, right? And we also need to be able to process information. So instructions that enable the cell to process the matter and energy have to be interpreted by other cellular systems. So it's a huge, it's like a big game of telephone. I don't even know if you guys even know that game of telephone, but anyway, it's a huge game of telephone as information is being passed from one system to the next. So that needs to happen. Now, besides that, cells are also going to do a few other things, but these aren't necessary or necessarily necessary for survival. They're going to reproduce. Okay, so the information that runs the cell gets passed on to next generations of cells. Part of that cell theory. Okay, cells beget other cells. And they also need to be able to communicate directly with other cells. And we have systems for that. Well, our cells have systems for that, right? So let's go inside the cell. So first we're going to start with the cytoskeleton. The cytoskeleton, that is the internal sort of structural, the matrix, the scaffolding that holds the cell together and gives it three-dimensional shape. This is structural proteins. They extend throughout the cytoplasm. It's a really, really important piece. You can see how when they're stained green and orange, you can see how they run throughout the cell in a fairly organized manner that allows for them to do that. This is where the MTOC comes into play. The microtubules that make up the cytoskeleton, made of tubulin. You've got microfilaments of actin, intermediate filaments. These are all things that work in conjunction to create this, like I said, that scaffolding that's going to hold everything together. And all of these are made out of protein. So protein is what drives this. The functions are support, maintaining the shape of the cell. It anchors organelles to the cell so it works well. You get regulation of the cell and organelle motility, so the movement of organelles around the cell as well as how they're arranged and organized. And the important piece, they are what make up spindle fibers that are going to move chromosomes during cell division. Okay, so that's how they work. So you can see motor proteins that connect to the microtubules are going to help move vesicles along them. Check that out. How does a vesicle move through a cell? It actually runs on tracks. Seriously, how cool is that? And what's going to make it move? ATP. Energy is going to help power it across. And you can see that right there. That's cool. You also have the actin-myosin connection that work in conjunction with muscle cells. The ability for amoebas to move around, okay? That flow of cytoplasm, okay, is really important. And then you have the movement of chloroplasts within a cell is actually driven by this as well, and actually move along these microfilaments to organize and move in the best way in which to capture light. So how pretty is this? So it's really, really cool. It's a regulator of cellular structure and function. It helps anchor and move, and they can actually stain them, and they're so pretty. Cilia Flagella. This is a extension of the cytoskeleton protein. So again, an extension of that MTOC. This is specific to cellular movement. And it's going to help cause motility. So flagella there's one human cell type that has flagella, and it's for movement. So, yep, those little swimmers, need to get to a particular place. So it's the flagella that's going to move back and forth in order to get them there. Cilia are usually almost like hair-like extensions around the whole surface of the cell, and they'll beat in order to move something. So like the cilia that line our trachea and our bronchial tubes. They're made up of the microtubules that make them up are organized into basically this 9x2 organization where you can see 1, 2, 3, 4, 5, 6, 7, 8, 9 around the outside with 2 on the inside. Sometimes you have it in a triplet formation, and this is what makes up the ability for them to move in that sort of, I guess, rod nature that gives it. Function, motion, that's what they're there for. The ability to move through space. So here we can see sperm cells all covering an egg. You have things like paramecia, little single-celled protists that swim through ponds by beating flagella back and forth. So let's get to the actual division at the cell level. So at the boundary, we're going to have the cell membrane. You guys have some pretty good insight into cell membrane. So let's talk structure. Fluid mosaic model. It's a bilipid layer of phospholipids, associated proteins, and a few other accessory type molecules that are in there as well that we'll talk about. Everything from cholesterol to other glycoproteins and polysaccharide chains that are going to stick out. Okay, so you got this gorgeous double layer that's going to be there. So you can see that if you were to kind of pull apart, and they've actually done this, which is kind of crazy, you basically freeze a cell and you can actually pull and open up that lipid bilayer and actually pull it apart and see all of the accessory proteins sticking out of it. So that's how, part of the way that they knew that there was this sort of fluid, this mosaic model where you've got like all of these proteins just kind of shoved all over the place, which was really cool. So the functions, they're the boundary. It's the bouncer of the cell. It's going to regulate what moves in and what moves out, transport of materials, communication between the cells. This is the, everything that happens from the cell receiving signals to mobilizing information to moving stuff out of it, all is regulated at this point. So you can see kind of the breakdown. So the pieces of it from your carbohydrate chains that act like little receptors and sort of like little antenna that are going to help determine cell, like self versus non-self. You have glycoproteins that are little glycogen extensions off of a protein molecule. Globular proteins, protein channels, the spanning receptor proteins that are there. Okay, all kinds of things that make up this fluid mosaic model that is our cell membrane. So huge amounts of types of proteins. So the phospholipid bilayer, piece of cake, right? We know phospholipids at that part. There's our little dancer. Hydrophilic head, okay, water attracted. Hydrophobic tails, boom. They don't like water. They're going to be repelled by it. So the membrane phospholipids organize themselves in a way where the phosphate is attached to a glycerol, has the 2 fatty acid tails, the polar your hydrophilic head faces either inside or outside the cell, while the tails scoot their tails inside, and there you have it. Okay, and they will spontaneously organize into this manner in the presence of water due to this dual nature of being partially polar, partially nonpolar. But they are not bonded directly to one another. So what that means is they're constantly moving around. But because of this dual nature, it always will maintain that barrier. I mean, seriously, how neat, right? Okay, so because of this, in this dual nature of these molecules, it will actually create a selectively permeable membrane where only small nonpolar molecules can easily through the phospholipid bilayer. Okay? So why would that be? Why would those only be able to go through? But in some cases, you can have some polar ones can go through, like water can. So let's look at the role cholesterol plays. They are one that's in there. When we have that addition, okay, unsaturated versus saturated hydrocarbon tails, right? We have our little kinky that happens, right? So when you have unsaturated or too many unsaturated ones, you get tons of space, okay? When it's super viscous and real thick, okay, it's going to be real tight. So then you get cholesterol that gets shoved in there, and it acts as a temperature buffer. It helps maintain the membrane fluidity over wide ranges of temperature, okay, so that we don't end up having cell membrane breakdown. It also allows for a little bit more rigidity for that cell membrane as well. So the membrane proteins, these are biggies. There's tons of different types. You get various depending on the job that they have and the role that they play. But you can pretty much break it down into two types. Either it's an integral protein, which penetrates either one or both layers of the bilayer, or you have a peripheral protein. And what that is it's going to be associated with the membrane, but it doesn't span the actual lipid bilayer itself. So here we have kind of the big picture, all right? So that's why I'm not even in this frame anymore, because you just need to just soak it in and take it for what it is. You should be able to recognize the parts, be able to understand why they're there, and be able to explain that within that cell. So here are some functions of some of the more typical membrane proteins. You should be able to understand them, recognize what they do, and so on. But there's a ton, but let's just quickly run through it. Transport proteins, they are what they are. They move things across the lipid bilayer that normally can't make it on their own. Some are passive, some are active. Enzymes are actually going to sit in the lipid bilayer as well and do their enzymatic activities right from the middle of the lipid bilayer. We're going to get receptors. Every hormone receptor is a protein that sits in the lipid bilayer, and the hormones come in, attach, cause some form of reaction that occurs. They can be single spanning, they can be 7 spanning. So there's different forms of these receptor proteins. You have cell-cell recognition pieces, so your glycoproteins that have these polysaccharide chains that come off of them. That's going to help one cell recognize another, so it's important for cell-cell recognition. You have intercellular joining proteins, and this is going to help create junctions between two cell types. And then you have attachments to the cytoskeleton and the extracellular matrix. Okay, so what's going to help hold a cell in place to what it needs to be held onto, right? So let's take a quick little spotlight on membrane receptors. I guess I have a little bit of a soft spot for hormone receptors and hormones. It's probably, to me, if you can understand how hormones work and how they respond at the cellular level, you can have a really solid understanding of cellular function, how cells work, and with structure as well, because things don't happen in the cell without a signal coming in, and that's how it works. So these membrane proteins create that that catch for these extracellular and intracellular signaling mechanisms. So they always have sort of the N terminus, they have the C terminus, so where signals come in, where they go out, and they create that signal transduction, that movement of that message from wherever it's coming from to whatever it has to do for the cell to respond. So something like this. So membrane receptors, though, can be used and abused by other organisms, such as viruses. So for example, HIV. HIV attaches directly to the CD4 receptor on the surface of our helper T cells. In order for it to do what it needs to do, it literally has to dock to that surface of the cell. Okay? So it infects the cell that has the CCR5 on its surface, okay, along with the co-receptor. So it's actually not only the receptor, but the co-receptor that it actually has to dock to. Okay? Then by docking, it can attach, and then it shoots in the RNA into the cell, and the cell's infected. However, there are some people who are missing the CCR5, so they are CCR5 deficient, okay? As a result, this is just amazing. As a result, HIV cannot infect the cells. So people who are CCR deficient, in essence, are immune to HIV because there is no mechanism for that virus to attach onto, to dock, and to then send its message inside the cell. So let's take a look at glycoproteins and their job. Structure, okay? Integral proteins, they span the bilayer, short polysaccharide residues that sort of erupt from the top, right? Their function, cell-cell recognition, where glycoproteins serve as an identifying marker, okay, to other cells in the population, like to others of your cells. So that's their job, right? So glycoproteins are really important for that. Okay, and they're a complication for organ transplants. Okay, so if you get a new organ, one of the reasons why you might have rejection of that organ could be due to the fact that there isn't the correct glycoprotein match between your cells and your new organ. So that is just one of the complications that can happen. So kind of think about that a little bit. So with that, I'm going to end right there, and we are going to continue our discussion in the next podcast on what happens outside the cell with other types of boundaries and barriers and ways to kind of separate the inside from the outside. So there you have it. Go back, check things out as you can, move through things that you feel like you already know and feel strong about, but that's where it is. So we'll see you in class. Have a great night, guys.